For decades, the standard view of cancer has been the somatic mutation theory. This idea stems from research in the early 1900s suggesting that cancer is primarily a genetic disease caused by random mutation in a cell's DNA. It's a view so dominant that if you ask most people what causes cancer, they're likely to say genes or environmental factors. But what if this is only part of the story? What if cancer is not a genetic disease at its core, but a metabolic one? My guest today, Dr. Tomás Duraj, is here to challenge that conventional wisdom. He and his colleagues are at the forefront of a paradigm shift, viewing cancer as a mitochondrial metabolic disease. This emerging theory suggests that cancer cells are fundamentally different from normal cells in how they produce energy. While normal, healthy cells are metabolically flexible, cancer cells become dependent on a less efficient form of energy production, relying on high rates of glucose and glutamine fermentation. This is known as the Warburg effect, an observation which earned Otto Warburg the Nobel Prize. Dr. Duraj and his team are attempting to leverage the Warburg effect with a powerful therapeutic framework that exploits this metabolic difference: Ketogenic Metabolic Therapy, or KMT. KMT is a strategy that combines a low-carb ketogenic diet, which shifts the body's energy source from glucose to ketones, with other therapies to target cancer's unique vulnerabilities. Today, we'll dive deep into the history of cancer research, the flaws in the current paradigm, and the exciting potential of KMT. Dr. Duraj will explain how this approach is not just a diet, but a comprehensive strategy to create a metabolic disadvantage for cancer cells while protecting and empowering healthy ones. You'll learn why this perspective is offering hope and a new path forward to fight against cancer. Join me in vivo with Dr. Tomás Duraj on challenging the cancer paradigm. So in terms of what you and your lab study, I think to lay some groundwork down first, sort of as a first principles thinking, the current paradigm in terms of the idea of cancer really stems from Theodore Boveri, 1914, the somatic mutation theory of cancer. I think if you were to ask anybody on the street, where does cancer come from? They're going to give you two answers, either mutations or environmental factors. So before we get into what you and your lab are exploring, why don't you tell us a little bit about Boveri, the somatic theory, and where most of the culture lies when it comes to oncology right now?
Yeah. I think maybe we could do like two sides, like two tales of cancer research. Maybe I could sort of explain a little bit the history of the somatic mutation theory, and then as a counterpart, the mitochondrial metabolic theory, and there is some other ideas around that we could also discuss. When we think about what cancer is, I would probably leave that to the philosophers of science. I think in terms of sort of the phenomena or the observation, cancer is just cell division and out of control in a multicellular organism, and I think that can also give us clues about how we define cancer, what the origin of cancer might be. And I also realize we're going to spend a little bit of time sort of explaining the history, or going through the history of cancer research. But I sort of know that from the work we do, most patients, when they are either thinking about prevention or treatment, they don't really care about that much.
Yeah, that's true.
But
good point.
So we should also try to spend a little bit later some time, going through like the practicality of how do you apply some of the concepts that we have been developing in the clinic. But to be honest, I think if this is like a scientific literacy issue, I think it's absolutely necessary to sometimes understand, maybe not the whole history of cancer research, but just a little bit of, as you said, like how is cancer research or investigated in the lab or in the clinic to understand, why do we have the treatments that we have, or the approaches that we have. So anyway, the somatic mutation theory, before the 19th century and 19th century, beginning of the 20th century, there was a little bit of debate still about the origin of cancers. There was different ideas floating around, the viral theory. Some people, and to this day, think that it's like a developmental embryogenic sort of failure, the morphogenetic fields or the tissue organization field theory. I think, with the work of Theodore Boveri in 1914, this was actually one of his later papers, I believe, because he died in 1915. he was looking at sea urchins, and more studying the gametogenesis, so the sort of the chromosomal numbers and chromosomal alterations that you could see in during development, and he sort of postulated that cancer might be sort of a defect in chromosomal sort of number or chromosomal structure. And there were other people. There was Hensman. Other people were sort of taking these ideas, but this wasn't defined yet as somatic mutation theory. I think that came later with Karl Bauer and Ernst Stizzer in the 20s and in the 30s, where they said, well, yeah, if cancer usually arises in the somatic tissue, which is sort of the mitotic, post-embryogenic tissue, then cancer could be defined as a somatic mutation, as a problem in the sort of DNA, or that wasn't discovered yet, problem in the genetic material of the cell.
Yeah, just a little bit of context, like you said, DNA wasn't discovered yet, and obviously the idea of genes and genetics was not necessarily fully known. It was postulated here and there, Mendel, right? But the idea of mutations of genes and what genes are was not yet available.
Yeah, I encourage also people to read maybe, the Carlos Sonnenschein and Anna Soto here at Tufts University. They sort of propose this tissue organization field theory, but they have done an excellent job going through the history of the somatic mutation theory and describing sort of the flaws of it and the historical development. So I think that could be a good, they have a bunch of papers on this, and they could be a good reading sort of experience to see how we got where we got. And so yeah, as you said, the DNA structure, of course, with Watson and Crick and Rosalind Franklin and many others that work on characterizing the structure of DNA in 1952 and 1953, this sort of sedimented the idea that there could be changes in DNA structure in the information contained in the DNA, and this also sort of coincided with models of carcinogenesis, so like different development on when you apply a carcinogen, like a chemical carcinogen, like 20-methylcholine or tar, and you could sort of with certain degree of replicability, you could induce tumors in animals, in animals of experimentation, and you could say, well, we see this connection where we apply this carcinogen, then we try to see what changes occurred in the tissue. There's inflammation, there's DNA damage, there's also mitochondrial damage, and you could postulate that cancer, based on this retrospective observation, you could postulate that cancer could be due to defects in the DNA, which indeed are observed. Now, I think that the question is, how many defects would you need to make a cancer cell behave like a cancer cell, genetic defects? There's also different perspectives on this. I don't know, Carl Nordling, he was more looking at the epidemiology in the 50s, and he proposed that, based on how cancer is more incident, more frequent in the later stages of aging, then perhaps you would need 7 mutations. That was sort of his calculation based on just mathematical modeling. And David Ashley also in 1969, he said, well, maybe it's 3 to 7 mutations. This is a multi-mutation theory where, yes, we now know that there is defects in DNA. And how many do you need? Well, maybe 1 is not enough. Maybe you need a couple more. So they said maybe 3 to 7. And this idea actually sort of came back into view later on. Now we have the idea of driver mutations and passenger mutations. But given that this framework sometimes doesn't work, that you can artificially increase the number of mutations that you would need to develop a tumor, post hoc. So today with, for example, polyps, you can see that one mutation is typically not enough. So there's this idea that you need several mutations in different pathways. There's the two-hit hypothesis by Knudson, who would describe basically tumor suppressor genes in the retinoblastoma gene where you could inherit sort of a predisposition factor to cancer. We could also talk about that a little bit. And later on, now we're getting to the 80s, the 90s. This is the discovery of the oncogenes. Varmus and Bishop and Robert Weinberg and many others, for example, describing the carcinogenic potential of the RAS oncogene, which is sort of very famous. And this is really where I would say that the somatic mutation theory was sort of solidified, because you could take a virus transfection or a transduction or a transfection method, and you could insert oncogenes or inactivate tumor suppressor genes in normal cells, and you could observe that you can develop tumor cells. You can have tumor cells. You can have cells that acquire tumorigenic potential in the sense that they now make a tumor in a mouse model, and this sort of solidified the idea that if we target the DNA, we can insert a gene that is dysfunctional, that is overactive, and we can observe that you get a tumor. We have some of course different ideas about this, because all of these oncogenes, the KRAS, the MYC oncogene, and many others, they localize in the mitochondria, and they also are involved in mitochondrial metabolism. So you are sort of doing two things at the same time. You are inserting a gene that signals proliferation, but at the same time, this has to be mediated by mitochondrial function. And in sort of current perspectives, I would say we have by Greenman, we have the idea of driver genes and passenger genes. We have the Cancer Genome Atlas, which sort of sequenced a long sort of database of tumor samples, and 20,000 tumor samples, I believe, across 33 different types of tumors. And of course, we have detected thousands and thousands of mutations. And the bad luck hypothesis also postulates that perhaps you cannot do much to prevent cancer, and this has been heavily criticized since Thomas Duraj and Augustine, which basically said that the more replication you have in your cells, that the higher the predisposition to cancer. So I think there's right now within somatic mutation theory, there is the perspective that cancer can be random, so you can acquire random mutations, and those are not related to the exposure to external factors. And of course, there is also external factors that make sort of DNA damage perhaps more frequent, or more increase the predisposition to that. So that's sort of a little bit meandering, but that's sort of the history of the field that I would give so far.
Right, and you mentioned, and we'll get to this later when we speak about what happens in the lab. Excuse me. When you mentioned MYC, for example, transfections, this is the idea of like you mentioned, ectopically expressing a gene in a cell line and then seeing what happens. But you mentioned it localizes to the mitochondria. My point is that in molecular biology, sometimes you've got too many variables, right, and it's very hard to keep one steady and alter the other, or to consider all variables at the same time, and is part of the problem when it comes to the somatic, the gene somatic theory of cancer. So we'll get to that a little bit later. Now you mentioned, and I was gonna ask you, what got you doubting these, and you outlined it in your paper very well in 2024. We'll link it below. You mentioned sequencing, the oncogene atlas, or the atlas, 20,000 samples over 33 different types of tumors. Despite the fact that we have all this sequencing, it's been very difficult to develop treatment. So this is one point of contention against this somatic theory. As well, you have all these samples, but you can argue that mutations are neither necessary nor sufficient to cause tumors, and there are examples of that. Could you expand on those two ideas a little bit further? Because you outline it in your paper very, very nicely, and I think it's important that people understand these 2 points.
Yeah. So, regarding the Cancer Genome Atlas, I I do admit that this is like a marvel of technology. The Human Genome Project and the Cancer Genome Atlas, it's like a very impressive, international collaboration. And the problem, I think, the idea was that we would be able to sequence different tumors, and we would be able to identify, sort of like Knudsen was saying, like identify specific mutations that would drive tumor growth, and we would be able to perhaps design targeted drugs or fix the genetic problem to try to solve, try to inhibit tumor growth. But after all this sequencing effort, what you basically saw that you had like intratumoral, intertumoral and interpersonal heterogeneity across all the cells. You have different tumor cells that have different mutations within the same tumor. Different tumor types have different, driver mutations and different passenger mutations, and different people have different mutations also. Of course, there is a common core of mutations that are predisposing, that are, I would say, they are mediating the malignant transformation. But I don't think within the heterogeneity you could infer like a causal effect. And when we talk about whether mutations are necessary and sufficient, I think there is some evidence that would argue against that. Of course, you have some tumor samples that have no mutations, but you could maybe postulate that is because, I don't know, the sequencing is not precise enough or not deep enough. The idea now is also that these mutations might be in the non-coding regions, so in the dark genome, which might be, but I think that's sort of trying to fit the sort of adjust the hypothesis and the model to fit the data, not the other way. There's also many oncogenes and tumor suppressor genes that can be active or inactive in normal tissues. So how do you explain if you have a p53 mutation, a KRAS mutation, an overactive MYC in normal cells that you sequence, and yet you don't see malignant transformation? So this could also be sort of argued away if you could say, well, maybe it's not the right order, or maybe in this case there is some sort of genetic silencing that makes these proteins not actually defective, even though you have the mutation. And then third sort of point of evidence would be the nuclear cytoplasm transfer experiments, which maybe we could also expand a little bit more upon. But these basically, these were experiments across different groups, but mainly Israel Schaefer in the 87, 88, where they took the cytoplasm, the karyoplast, so the cytoplasm of a malignant cell and the nucleus of a normal cell, and vice versa. They inverted sort of the combination to see whether cancer would develop, and they had some interesting results too. So this would be in my mind sort of the reasons why we would doubt the somatic mutation theory. I also think, like, on a practical level, unfortunately, I feel this was a very good idea at the beginning, and you can see the trail of evidence that made people and researchers focus on exploring this. And I think we have explored it sort of as much as possible. But in the clinic, if you look at sort of the approved drugs, by the FDA in the last 20 years, the increase in survival is very, very, mediocre, I would say, very poor. So just this approach of developing new drugs against genetic targets is, simply appears not to be working. Now, of course, you could say, well, maybe it's because of the heterogeneity. So I guess we have to work harder and develop, specific drugs to all the possible mutations that you can have in a tumor. But I don't think this will work, because you also have normal cells to worry about, and this was always the problem. How do you target the cancer cells while preserving the function and the health of the normal cells? So
it's important to also acknowledge some of the successes, like we have HER2 positive breast cancer with Herceptin or CML, Gleevec. But it's a very good point. Despite the fact that we were able to sequence all this, we haven't been able to progress treatment based on that sequencing as far as we would have liked. Now, you mentioned earlier MYC So this is a gene that produces a protein which is very commonly mutated. I think perhaps the most commonly mutated next to P53, or maybe it's the other way around, in cancer. This is a protein that I studied myself. You mentioned that it translocates, or it ends up in the mitochondria. So my question for you is, tell us about how the mitochondria and mitochondrial function is important in malignant transformation, and some of the strange things that we see in the cases of predisposition with MYC, KRAS. Ultimately the question is, does this suggest that genetics plays a secondary role to metabolism, which is where we're going to go with eventually with your theory, the metabolic nature of cancer. But talk to us about mitochondrial function in cancer.
Yeah. So as I said, maybe we would now tell the other side of the story. So we have Dr. Warburg, who is often credited with sort of studying the somatic mutation theory, and a whole host of very smart people that work on this perspective. But we have also other theories or other perspectives on how cancer might have developed. So from the metabolic side, from the mitochondrial metabolic theory perspective, we could tell the story of Otto Warburg. Otto Warburg got the Nobel Prize for his work on respiration, or on oxidative phosphorylation of normal cells and cancer cells, for the cytochrome C enzyme, the respiratory enzyme. He also described the nucleotides, so the cofactors, the reducing equivalents, FAD and AD. And in 1956, he published this sort of seminal paper where he was arguing on the origin of cancer cells, where he was arguing what is the actual primary cause or initial insult that you observed in cancer cells for cancer cells to behave as cancer cells. And he postulated that this could be due to respiratory injury, he called it. So that this is, in that time, the mitochondria were not even fully characterized. These were the grana, so we couldn't even see but it was known that within these grana, which are mitochondria, this is where oxidative phosphorylation is occurring, which is the generation of ATP, more efficient generation of ATP in the cell. But when he was working on cancer, he observed that cancer cells display fermentation, so lactic acid fermentation. So cancer cells in that time from tissue samples, from spontaneous tumors, he observed that these cells were fermenting, were producing lactic acid despite sufficient oxygen, even in hyperoxia. So he postulated that maybe there's something wrong with the respiratory apparatus of the cell. This was both in vitro and in vivo. And he postulated that this might be sort of the underlying problem, the metabolic problem that cancer cells are sort of driven by.
So this is a very important point that I just wanted to clarify for lay people: the idea of oxidative phosphorylation and cellular respiration, breathing. So mitochondria using oxygen to produce energy. Just, from a micro to macro, it's almost the same idea. And that you can think about it as healthy cells. Correct me if I'm wrong. So healthy cells, you can think about them almost like hybrid cars. They have the ability to burn or to create energy, burn energy in an efficient way as an electric kind of car, but then they can switch to a more, a less efficient gas version. But you've got these cancer cells that are stuck in this inefficient sort of gas guzzling way, which is producing lactic acid through glycolysis and that sort of thing. So it's just important to understand that cells can go down two paths when it comes to energy creation and burning, and that there's the Warburg effect. Dr. Warburg noticed that cancer cells shift towards this inefficient, we can call it fermentation, just like we see when you're brewing beer, producing lactic acid. Just want to get that out of the way because it's sort of a complicated idea. Metabolism is always complicated for first year students, but lay people, let alone, it can be very complicated. But I just wanted to lay that out.
Yeah, I think you're absolutely right. So I think the problem here is that these two things are present at the same time, and you can measure both.
Yes.
in the cell, in your cellular model, or even in the tumor that's growing in the animal. So this actually got Warburg into trouble because, in his initial work, he was also working in sea urchins and proliferating sea urchins. He observed that they normal cells, that they consume more oxygen. So he said, oh, okay, even proliferating cells are driven by oxidative phosphorylation. And then later in the 20s and in the 30s, in his work on cancer, that's where he detected this lactic acid fermentation, this sort of more inefficient way of energy production, which he tried to sort of argue in the Mayer quotient that the oxygen consumption was always lower, or slightly the basal oxygen consumption was always lower in the cancer cells. And I think this is not exactly the case. I would say oxygen consumption is highly variable, but it is not an actually accurate or meaningful sort of measurement of oxphos. You can have different cells with different levels of oxygen consumption, but it doesn't tell you how efficient is their actual energy production. It's very hard to like measure actual ATP, the energy currency of the cell. You can measure the total amount of ATP that you have in a cell, and then you can extrapolate sort of, oh, I see it very upregulated glycolysis, the enzyme myrokinase pathway. Okay, so energy must be coming from there. Okay, I see very upregulated oxygen consumption. Okay, the energy must be coming from there. But we don't actually, you cannot know. The energy, all you know is that if the cell has energy, and it's alive, it has to have energy to be alive, but it's sort of very hard to extrapolate. So Warburg, there was like this big polemic after his paper came out in 56 with Sidney Winehouse and other researchers, Herbert Crabtree, other contemporary researchers of that worked on metabolism at the same time as Warburg, sort of criticized him, saying like, hey, we can detect oxygen consumption, and we can measure with labeling sort of isotope tracers. We can see that, for example, glucose is also partially oxidized, even though a lot of it it's coming towards lactic acid fermentation, or fatty acids. They're also, to a certain degree, they are oxidized, even though maybe not as much as in the normal cells. But I don't think this really changes the thesis, because Warburg himself, and I sort of use the same term, he said respiratory insufficiency or oxphos insufficiency, which simply means that oxphos by itself is not sufficient to maintain the bioenergetic sufficiency, the ATP sufficiency of the cell,
right.
And it forces the cell to upregulate substrate level phosphorylation or fermentation. Now, as you know, I prefer substrate level phosphorylation because that actually talks about ATP energy. Right. Fermentation itself is simply usually defined by the lactic acid that you can measure in an assay. It doesn't necessarily tell you where the energy is coming from. But I think Warburg had the right idea. He has this very nice quote that I like a lot from his Lindau lecture, the Nobel laureate lecture in 1966. If I maybe I can say it, but so he said, like, when respiration disappears, life does not disappear, but the meaning of life disappears, and what remains are simply growing machines that destroy the body in which they grow. And I think this sort of encapsulates exactly his theory, and this is sort of the same idea that we are trying to postulate today, is that when oxidative phosphorylation, which kind of maybe you could get out also into sort of the atavistic framework and explain a little bit the evolutionary history of life on Earth and how cells have developed, and after the endosymbiotic event, that's where we can get multicellular eukaryotes. So if you lose function in oxidative phosphorylation, it's only logical that you would start behaving more as a prokaryotic organism, and you would upregulate glycolysis, which is the primary energy source of cells without mitochondria, of course. And all the cells that, prokaryotic cells, they typically proliferate out of control in their environment as long as they have fuel. So, right. So I think this is sort of the crux of the argument that we would be postulating. We don't think it's the somatic mutations. Somatic mutations are detected. They are likely the consequence of reactive oxygen species through to the mitochondrial dysfunction, which is sort of a very inaccurate term that we shouldn't be using, but loss of efficiency in the mitochondrial, in the chondriome, in the collection of mitochondria within the cell, and oxidative phosphorylation. The cells have a lack of energy, not enough energy to whatever their energy requirements might be, of course, during proliferation. And this is also a little bit debated whether they need more or less. I am of the perspective, from a physics perspective, that you need more energy to proliferate, to make new stuff. You usually
expect more. Makes sense.
Yeah. And so if there is not enough energy from ATP from oxphos, you must upregulate. You have to get it somewhere, otherwise the cell would die, or it's not able to fulfill its functions. So you upregulate substrate level phosphorylation, and as a downstream consequence of this, you start sort of expressing or behaving as a prokaryotic sort of cell, as a cancer cell in terms of multicellular organism, where you want to integrate the cell into society of cells. You don't want the cell being rogue and just proliferating out of control. So
allow me to summarize just a little bit the observations that you're making, that Warburg made, is essentially we see that cancer cells shift towards this inefficient because of oxphos insufficiency. They switch to this different pathway in terms of energy. Now, my question would be, you used the term initial insult earlier on. What is the initial insult that's causing this mitochondrial dysfunction, which you said is not a good term, but I'll just use it for now. What causes this switch? Because you also mentioned tumor heterogeneity or mutations being a result of this switch.
Right.
So what is that initial insult?
Yeah, I think this gets into the oncogenic paradox that Albert Sangiorgi sort of also noted in the history of cancer research, which is like, how is it possible that, if we look at the somatic mutation theory perspective, you have some carcinogens and some, insults, as we would say, that do cause mutations. In the Ames assay, you can detect, this definitely is mutagenic, and it increases the likelihood of cancer developing. But then you have other stuff, you have hypoxia, you have inflammation, you have different infections that do not necessarily cause mutations, but they still can cause cancer. And there's different, ways that you could fit it into the somatic mutation theory. But we think like many of these non-mutagenic insults, they can still damage mitochondria, either structurally, so you can have crystallysis, you can have defects in cardiolipin, in different lipids that are involved in the electron transport chain. You can also lose the efficiency of the function. So problem with oxidative phosphorylation in terms of like efficiency is that it involves many steps. So you have to get different fuel sources, get into the cell. They have to be transformed with different enzymatic reactions, get into the TCA cycle, the Krebs cycle. There's also many probabilities of having like an inefficient TCA cycle through different fluxes and how the different metabolites come in and the enzymes involved. And then you have the electron transport chain, another possibility to have inefficiencies. So you can lose function if the structure is abnormal, or if it's damaged, you can lose functional sort of efficiency. There's also mitochondrial dynamics, fusion, fission, mitochondrial number. All these things, I think, could be sort of the underlying defects that make oxidative phosphorylation less efficient than the cell requires. The problem is this is like not as elegant, I would say, as the mutations. The mutations, of course, it's like on off. It's like, okay, we are comparing whatever normal is sort of considered. Of course, there's some issues with like polymorphisms but whoever doesn't have cancer, you could say has normal so DNA content, and then you compare it with the cancer cells, and you see, okay, I see many mutations. So therefore, these must be the causal agents. Whereas we would say that it is this metabolic switch is actually the underlying. It's mediating the malignant transformation, but it also underlies the carcinogenesis to primary cause.
So you outline a very interesting fact in your paper, and then it's the fact that no cancer thrives without glucose or glutamine present. No known cancer, no known sample has been seen in the lab or in situ in a person. What does this tell us about the metabolic theory of cancer?
Yeah, I think that's more related with the efficiency of ATP production. So we have this running joke where we like offer like a prize to any researcher that could show us basically a cancer cell proliferating out of control using mainly or only oxidative phosphorylation. This is very difficult because even though I think normal cells can proliferate using mainly oxidative phosphorylation, you still need the metabolites. So this would have to involve like some genetic engineering, because you would still need to provide the glucose and make it not go into lactate and be fully oxidized. But we know that many normal cells, the regenerating liver and normal epithelial cells, even though they might slightly shift the ratio towards lactic acid production, and this comes mainly from like in vitro studies, which is a little bit difficult because you also have the Crabtree effect. Like any cell in vitro will have, most cells in vitro will have the Crabtree effect, which is lactic acid production, even though their respiration is normal. It is just an issue of the concentration and of glucose and a competition for phosphate and whatnot. So it's a little bit complicated, but I think we can say with certain degree of certainty that I think this makes sense just even conceptually. All we are saying is that we think that the cancer cells are more inefficient in their metabolism, in their bioenergetics specifically. Now, you could argue that cancer cells have a proliferating advantage in the sense that they can outcompete normal cells, non-proliferating normal cells. If you compare a non-proliferating normal cell with a proliferating tumor cell, you would say, oh yeah, proliferating tumor cell divides faster than the non-proliferating normal cell. So that's the advantage, I think. Even from the somatic mutation theory perspective, if you consider that there is damage to the genetic, so the fidelity and the function of the genetic blueprint of the cell, this is unlikely to confer sort of fitness, especially from the metabolic perspective. It could confer fitness if you are selecting clones that are more adapted to grow under hypoxia, or they can evade the immune system. So you can tell this story backwards, say, okay, I have these tumor cells that, maybe display invasion or metastasis, or they survived in a hypoxic niche, and they have these mutations which make them survive in that environment. So you can say, oh, that must be that the mutation is sort of conferring them the ability. But no matter how you look at it, I think if the more damage you have to both mitochondria and DNA, this will make the cancer cell more bioenergetically inefficient. So this is where we think there could be a therapeutic sort of approach.
So, hearing you talk about this, and I'm thinking back to my days in a molecular biology lab doing tissue culture, and with the idea that no cancer cell thrives without glucose and glutamine. As soon as I said that, I was reminded of media that you make up when you're plating cells. So for people who have never worked in a lab before, you've got these cells that are living and they adhere to essentially a petri dish, a tissue culture dish. And in order for them to survive, you've got to feed them with this media, and you have to make this media. And there's a couple of things you add in there: serum from cows, which gives it protein, amino acids, that sort of thing. Antibiotics, so nothing bad grows in this dish, but you also add glucose and glutamine.
Yeah.
Always. And if you
Absolutely.
don't, what happens? Your cells die, and it's a terrible mistake that you make in your lab because it costs you resources, but time to grow up these cells, and you have to start from scratch. And so this idea which is what you're going to get to eventually, is the ketogenic idea of restricting glucose. I mean, as anybody who works in a lab knows this already, whether they realize it or not, that glucose is required for cells to live. So
yeah,
I just wanted to put that out there.
Yeah, if I may, this actually ties very well into sort of my project in the labs I've been working the last couple of years. Hopefully the paper will come out at some point. I've been working on precisely this idea. So I've been very frustrated by the fact that, if you look at many experiments that are published, like most papers these days, not to be too critical, because we know it's extremely difficult to do research on science, but most papers have like very interesting, one or two very interesting phenomena observations where they describe something unexpected or something that is sort of interesting within whatever system they're studying. And then there is a lot of sort of mechanistic descriptive elucidation of why this phenomena might be happening. And then if I look at many different publications in cell culture that are trying to examine bioenergetics, and you can measure lactate, you can measure oxygen consumption, you can measure different enzymatic reactions. But there's always this problem where they are focusing mostly on short-term adaptation. So you can find many papers where they take fatty acids, and many fatty acids are also in serum. For example, dialyzed serum. If you are studying cells in cell culture, if you want them to grow, you have to provide serum. You have some fatty acids in there. Some of these fatty acids are oxidized. You can add ketone bodies. You can add lactate. You can have galactose. Many different oxidative fuels, or mainly oxidative fuels. And then you can measure your cancer cells, for example, at a short time frame. Let's say 24 hours. That sounds long, but in terms of cell culture, I would define it as a short-term experimental design. Yep. And you can observe, okay, I add ketone bodies. This is something that we get asked all the time because people are sort of interested. Okay, if I'm trying to inhibit proliferation of tumor cells by reducing glycolysis and switching over to oxidative phosphorylation with ketone bodies, will the cancer cell be also able to adapt? And you see invariably, if you add different fuels such as ketone bodies, not all the cells, because you require the enzymatic machinery to use ketone bodies, but some cells will see an increase, let's say in oxygen consumption. If you reduce glucose a lot or even to zero, and you add ketone bodies, within 24 hours, you see a big spike in oxygen consumption. And you could say, oh yeah, these cells are using ketone bodies. But then if you wait a little longer, in all of the experimental evidence that I've been able to find, and even in my own experiments, these cells usually die. So they are not able to sustain oxphos as a primary fuel, as a primary ATP source to cover their energetic needs. Now, is this because the mitochondria is inefficient? Is this because they have higher demand and they cannot satisfy the demand of proliferation with oxidative phosphorylation alone? Therefore, they require fermentation. That's an open debate, but I think this is a sort of a clear bioenergetic distinction between normal cells and tumor cells. And in my models that I've been able to work on like primary macrophages, if you stimulate these immune cells, for example, to proliferate using galactose and glutamine, they can proliferate using primarily oxidative phosphorylation. They proliferate slower than a cancer cell. This is regulated proliferation. It's not dysregulated proliferation. But I think across many different models that we have been able to see in the literature and even in our own work, cancer cells cannot sustain viability and especially proliferation using oxidative phosphorylation, whereas most normal cells, of course, you could argue that some cell in the body might be more dependent. Erythrocyte might be more dependent, but they need so little energy that maybe it's enough if you have just tiny bit of glucose. They can probably cover their energetic needs. But most normal cells can sustain viability, especially and proliferation too, using oxidative phosphorylation. So I think this is sort of the distinction that we would make. But I don't want to see more papers sort of doing these short-term 6-hour studies or 24-hour studies. When you see some change, then you do a whole genetic screen and metabolic screening, and then if you just do long-term like self-sufficiency or I call it ATP sufficiency. Like ATP sufficiency, if a cell is alive, you can extrapolate that any living cell must cover the demands of ATP necessarily. That's like a axiomatic statement. So if you see a living cell, you assume that it's covering its ATP needs, and then you can ask questions: how can it cover what pathways is it using? Is it OXPHOS? Is it substrate level phosphorylation to cover the APD needs? But if you do a short-term experimental design, you don't really know whether you have
You'll never see it
efficiency or not. So yeah, you can see many changes. You can screen. You can do a lot of genetic studies, transcriptomic studies, metabolomics, but you don't know anything about whether OXPHOS was sufficient or it's sufficient to keep the cell alive.
So I think this is a very good segue in. So let's, if we may, step back from the molecular biology and focus more on the clinical side and what you're proposing with ketogenic diets as a means of treatments. Now, specifically, you in your lab, in your experience, you've dealt with glioblastoma, which is historically has had a very, very low survival rate, 14 to 21 months. And it was only 8 to 14 months in about 1926. So it hasn't changed much despite all of this data, sequencing what we know about cancer. It hasn't changed much. So what have you seen? What are you proposing with the ketogenic diet, which I think most people by now have heard the idea of ketogenic diet. So maybe you can redefine that for us and what you're proposing as a treatment here.
Yeah. So I think also here we could take just a quick step back. Like, cancer metabolism is experiencing a big resurgence in the field. I think whether researchers state it or not, they sort of feel that a lot of the ideas of like targeting specific mutations have been like that avenue has been exhausted, and it's really not leading to results that would be deemed acceptable in terms of improvement of survival. So there is a lot of people working on cancer metabolism. The problem is right now in the clinic, you basically use metabolism only as a diagnostic tool. So with 18 fluorodeoxy glucose PET scan, you can see upregulated glucose uptake, I would say, because people get a little bullshit about this. Like you don't really know what's going on with the glucose technically.
Right.
You cannot call it Warburg effect because you don't know whether it's going to lactate, but you can do tracer studies, and yes, it's mostly going to lactate in many tumors. But you can use labeling or tracers to detect tumors based on their uptake of glucose. But clinically, you don't do anything with that information. There's like no approach
Very strange.
is standard of care to use this very clear metabolic difference. So I would say the thing that we are proposing is, basically use what we know about the underlying biology of cancer cells that display OXPHOS insufficiency and substrate level phosphorylation dependency, which is often accompanied by the Warburg effect. The Warburg effect is typically defined simply as glucose to pyruvate, instead of pyruvate being oxidized, it goes to lactate. That's sort of lactic acid fermentation. As I said, I don't like that term that much. I think substrate level phosphorylation dependency or increased substrate level phosphorylation flux is more accurate, because you are talking now about the pathway of glycolysis itself, the Embden-Meyerhoff-Pinus pathway, which is more active in cancer cells. And this is what gives you ATP. The lactic acid fermentation, the Warburg effect itself, which Warburg also never used. This is a term coined by Ephraim Racker. Doesn't tell you anything about ATP. You could have fermentation technically with absence of ATP. So right. This metabolic phenomena we think could be sort of exploited for therapeutics using ketogenic metabolic therapy. I also sometimes I have low preference to focus only on the ketogenic diet. We are not even a lab technically that is interested in nutrition, even though we are sort of by popular demand, we are talking a lot about dietary interventions in cancer, but we are more interested in the underlying cancer bioenergetics. And dietary ketogenic metabolic therapy in the form of a ketogenic diet or in the form of fasting is simply a tool that you can use in the clinic, and then that any patient, I would argue, can sort of implement, ideally with a dietitian. But I think the dietary aspects of cancer management are still sort of open to the patient autonomy, where you don't need permission from anyone to implement this, as long as it's done properly. which is a tool to basically leverage this metabolic inefficiency of the tumor. So you are reducing the availability of glucose. You are reducing inflammation. You are reducing angiogenesis. You are upregulating autophagy, especially if you combine ketogenic diet with transient caloric restriction or fasting, and you are improving the immune surveillance and the recognition of the tumor cells by the immune system. So all these beneficial effects you can induce by a dietary intervention, which happens to be a ketogenic diet. The holy grail, of course, would be like, okay, I design a pill to give the patient to mimic all the sort of pleiotropic effects of the ketogenic diet, but that will be extremely difficult. I would say impossible, because it changes hundreds of pathways and reactions, because you are shifting your body to basically be less dependent on glucose as a primary fuel. Your normal cells will now still oxidize the glucose, but much less of it. And you are upregulating oxidative fuels, not only ketone bodies, but also fatty acids as the primary sort of ATP fuel for your cancer cells. You still, not for your cancer cells, for your normal cells. You still will have glucose in the blood. This is another common criticism, like, oh, you do a ketogenic diet, your blood glucose goes from 5 to 3.5. There's still plenty of glucose, but I think this also misses, like, there is a lot of competition sort of in the environment to use that glucose, and your normal cells will be preferentially using that glucose over the cancer cells, especially if we accept that the cancer cells are more inefficient in their bioenergetic function. So you are reducing the glucose, you are upregulating ketone bodies and fatty acids and other oxidative fuels as a primary source for your normal cells, and you are putting pressure on the tumor cell to be basically less fit bioenergetically, to be less adaptive to this new environment. And this is just one portion of the strategy, as you mentioned in the paper.
Right. After
this should come, the actual targeting of glycolysis and glutaminolysis. We haven't talked that much about glutamine yet, but glutamine is another essential fuel for the cancer cells. We think it's contributing to mitochondrial substrate level phosphorylation to a certain degree. The degree is still under study, under research, but there is another way to make energy inside the mitochondria that does not involve oxidative phosphorylation, which is the succinyl-CoA ligase step in the TCA cycle. And we know, and nobody would argue against this, like the amount of flux of glutamine through the cancer cell is upregulated. And we think that mainly this pathway of glutamine to alpha-ketoglutarate, then to succinate, this is like very highly upregulated. Some of this succinate will be oxidized, but it's mostly similar to glycolysis, to the Warburg effect. Cancer cells require this other way of making ATP, which does not involve oxidative phosphorylation, to cover their ATP needs. So targeting glycolysis and glutaminolysis together with this dietary baseline that sort of is there not only to make pressure on tumor cells, but to also protect your normal cells would be sort of the strategy. I think we have done a lot of work on the first part of the ketogenic diet, and this has been studied for the last, I would say, 30 years, 20 years certainly in the Seafried lab. We have been studying ketogenic diet as a baseline. There's many clinical trials that have been done already. The results are heterogeneous, I have to admit, and there's many different explanations for this. One of them is also the implementation is a little bit difficult in a cancer setting. It requires a lot of supervision, and whenever we have like exceptional responses in case reports and case studies and case series where there is like more direct involvement with the patients. But whenever there has been like, okay, we're going to test the ketogenic diet in like a big institution, in like a big hospital center, in the clinic, we get into issues with like, the IRB and how do you design the diet so that the patient doesn't lose any weight. Therefore, you have to like overfeed the diet basically. And even on a ketogenic diet, if you overfeed a ketogenic diet in a mouse, especially, you can make the tumor grow faster because glucose never goes down. You never reduce, the signals to proliferation. So this has not been observed in humans. Apparently humans can overeat a ketogenic diet. Certainly it doesn't make the tumor grow faster, but it might have neutral effects. So some of these clinical trials have shown I would say neutral effects, neither acceleration nor deceleration, but preponderance of evidence, I think it supports the diet itself has anti-cancer effects. And the second part would be to add some pharmacology to target glycolysis and glutaminolysis together with care.
Yeah. So you mentioned, or you said that it'd be nice to make a pill, but it's tough to cover all these pleiotropic effects. And that's the wonderful thing about what's being proposed here is that it doesn't require a pill, that you can do it on your own, ideally with the supervision of a doctor. But I can tell you from personal experience, I have two friends who implemented these ketogenic diets because they were battling cancer, and completely anecdotal, both have had very positive effects. And the nice thing about a ketogenic diet is that in addition to sort of trying to fight cancer here, it's a very healthy lifestyle, very healthy diet, and both of them have had positive changes in that regard, lost a lot of weight in a very healthy way. They feel better, et cetera, et cetera. Whether they had cancer or not, I think they would both agree, I would have done this anyway had I known how good I would feel. So I think that's very, very positive. How has your work, because we know, if you zoom out, there's lots of battle going on between different vested parties. You have researchers, institutions, pharmaceuticals. How has your research been, taken? What sort of feedback have you received?
Yeah, I think this is a very different, interpretation that you see across different levels of the lay public, of other researchers, of clinicians, which are sometimes combative because they feel like you are infringing upon their sort of standard way of doing things, which it's understandable. It's a profession and it has its internal sort of way of code of conduct and way of doing things. And if the patients bring different ideas to discuss, I think sometimes there's no time to discuss. That's another problem. Within most, if you are going through the informed consent and decisions of treatment, it's so rushed because maybe you had a tumor for, I don't know, 6 months or a year before you detected it. But after it's diagnosed, you are sort of onboarded into the whole system and the procedure. The wheels are just turning and you are being diagnosed, you're being offered different types of treatments, standard of care, chemotherapy, radiation, surgery, maybe also thinking at that moment already, okay, what happens if we have to go to second line treatment? What do we have? Do we use a targeted therapy? Is there anything approved? I think this is one of the problems in clinical, in oncology, especially. I don't know if this happens in other fields, but I feel this was one of the reasons why I switched to a research sort of program or to a PhD. So I feel I could maybe help to give clinicians the tools that they could use in the clinic, because sometimes I feel you are treating the patient, you are thinking about okay, personalized treatment, but actually there's these consensus guidelines and you are doing a molecular diagnosis. Basically, you are, okay, we're going to take a biopsy or take a tumor sample. We're going to screen it and hopefully there will be a mutation, KRAS mutation, IDH mutation, ALK mutation, EGFR mutation. And many of these drugs are not really first line. So you want to even use it then, but you could have it in the back of your mind for okay, if it's not approved for this type of tumor, colorectal cancer or whatever, okay, I have it there. It will probably not work because I know there's heterogeneous mutations in there, but it might extend survival a little bit. So you're sort of waiting for the holy guidelines, consensus guidelines to for the researchers basically to give the tools after clinical testing, after clinical trials, to give the clinician the tools. And I think this it's sort of limiting. I don't think personalized medicine is here yet, even though we talk about it. I think the individual patient should be the focus of attention. There should be a shared sort of discussion. There's a lot of thought about shared decision making, but it's not really, in my mind, it's not really actually shared decision making. Like the clinician usually thinks by the training and has seen many other patients in the same situation, it's trying to provide them the best sort of tools that he can offer. Mm-hmm. But if the patient brings other ideas or wants to be like a proactive, like literally like sit down and read the clinical trials that support the standard of care, read the literature about other therapies that might be applicable that they might do on their own or with the help of the physician or dietitians or integrative, oncologists, or other professions. This doesn't really, I feel, it's not standardized. So it's like, you can seek it out, but I don't think it's like offered within the initial sort of onboarding in cancer. And this leads to the conflict that I was describing. Like, I think the clinicians sometimes can be a little bit, especially if they don't have time to deal with this, they sort of ignore the patient. Say, oh yeah, you can do diet. I wouldn't even say they discourage. Like most clinicians do not discourage the diet, but they say, I don't know what to say. Like they say, do it if you want to. But if you like started talking about, I want to try like a glycolysis inhibitor, I want to try deoxyglucose, or I want to try 6-deoxyriboxylmaleucine, DON, it's like a glutamine inhibitor. I've been tested to phase 2 clinical trials. Then they like roll their eye. They say, I don't know. Like they say, this is outside what I've been trained, and there is really no place to go. Now you would say, this is on us as researchers to like advance this as fast as possible, but that it takes like 20 years from like observation to drug in the clinic. So I think there's right now this disconnect where we know there is promising therapies, but the access to them is not yet implemented. And from the researchers, I would say, we haven't had that many like debates or conversations with others in the cancer metabolism field. I think this is more like a personal preference. Most researchers, I would say, and there's another problem, like when we talk about all these theories today, like somatic mutation theory, metabolic mitochondrial theory, there's tissue organization field theory, viral theory. I don't think most researchers, I don't know, maybe you could say what you think about this, but I don't think most researchers are thinking about like this grand unified theory when they are working on an individual experiment. You have like a hypothesis that applies to your model, and you are trying to answer the question that you might have like in your mind, like, is this, what am I expecting to see when I do this experiment? This should go up, this should go down within like very confined structure of the actual experimental design that you are working on. But I would say most researchers are not thinking about, does my result actually translate into like predictive power within the somatic mutation theory? Like if I do something and it's completely opposite to somatic mutation theory, do I still publish the result? Of course, it's like it's not connected to any real sort of application in most cases. Yes. And then there's individual sort of thinkers or like how do you call it, like prominent figures in the field that sort of shift the direction. It's constantly, there's Vogelstein, there's like different researchers that have been working on the idea of like mutations itself. And in our case, I think Thomas Seyfried, my mentor, and others are sort of thinking more on the metabolic perspective and are like building the theory around the observations. But most of the observations are unlinked to any theory. So I don't think there is hostility from other researchers. I think most researchers just don't know what we are talking about. And if you try to discuss with them, I think debating science is very difficult because like technically, or I would say impossible actually, on like a live stage, because technically they should be like, okay, let's go to this paper from 1956 or 1937. Let's look at figure 3 and let's discuss the experiment and how this might relate to other experiments. I don't think this can be like done on air in like a typical debate setting. So most of the interactions have been like, yeah, we think there is some point to the. We know there's like the actual experimental evidence for the metabolic changes is pretty clear, and then it's just a interpretation issue. Like we interpret this. It's the chicken egg situation. We interpret this as the mitochondrial oxphos insufficiency is primary. It's the initiating factor, and then there is reactive oxygen species and mutations accumulate as a downstream epiphenomena. And they would say it is the mutations. They signal proliferation, which upregulates substrate level phosphorylation flux. It could downregulate oxphos as an adaptation to this. And the experiments are the same, but the interpretation is completely opposite. As I said, there's the nuclear transfer experiments, which I think argue more in the way that the cytoplasm or anything containing cytoplasm, which we think is mitochondria, is the driving force. But you could try to like argue your way into different explanations. So
yeah. You've highlighted. So I'll speak from a molecular biology lens, which is you're coming from that lens as well. But you've highlighted a double-edged sword here, and I would agree with what you were saying in terms of what scientists, researchers are thinking about when they're doing experiments. So the double-edged sword is that without these very granular experiments, and when students ask me how do labs work, well, essentially a lab in molecular biology will focus and study one gene, one protein for the life of that lab. As long as the boss, the principal investigator is there, it's always just one protein, maybe two, maybe three, but it's very granular. And then it becomes very siloed. You have this lab is focusing on this protein, this pathway, that's it. And they're very hyper-focused on it. And the other lab is doing something different, but in a similar fashion. And they're never really thinking about the grand picture. And that really did bother me when I was, and I thought about it. I thought about this problem, but I too was not thinking, you're almost not allowed to think on a more macro level, because when you're in a molecular biology lab, it's very focused, as I mentioned. So you have to stay focused, observe, interpret the results based on the lens of that lab.
Yep.
So it does become very, very siloed. And another problem, I mean, if you're going to walk away with anything from this discussion, is that there are so many unknowns and there are so many levers that can be tweaked, but you can really only tweak one at a time and keeping others steady. And so it's a double-edged sword. Without these very granular experiments, we wouldn't know the basics behind biology.
Yeah.
But at the same time, you've now developed this problem that you've very beautifully articulated, is you're not getting crosstalk, and it's getting very siloed. And it would be very interesting if you had clinicians working in a hospital or any clinical setting where they had a molecular biologist on staff that they could tap when they're starting to deal with a case with the patient.
I think this is exactly right. I think this is also like science is made by humans, which is often forgotten. And when you're trying to publish, and there's a lot of pressure to publish, when you're trying to publish a paper, a manuscript with any data set that you have, humans are driven by stories. So usually you just pick the data that supports the story that you were going to tell anyway. I feel sometimes, and this, you have to be very cognizant of this to try to avoid doing that. So I would say, like, for any other researchers or even lay people reading the scientific literature, I typically encourage people to look at the actual data itself without ever reading. Like, most clinicians only read the abstract, and I would say most researchers, with the time we have, only actually read the words that the authors intended to be shared. But this is where it gets into the problem. For example, when I was talking about like ketone body oxidation in cancer cells, there's some labs that say there's no ketone body oxidation. There's some labs that say there is some ketone body oxidation. But you have to like do a deep dive in the actual data and say, okay, is this representative of the clinical situation when I would be treating the cancer with the dietary intervention or inhibitors of glycolysis glutaminolysis? So there has to be like this synthesizing effort, looking at data, not like figures, not works. And I think
yes.
this is
And
often,
let me decide as a reader. I will decide the veracity of your claims and not just take the abstract as you mentioned. And actually reminds me, you said, what's the story that you want to tell? Avoid that sort of method. I can tell you from my experience and from many other of my colleagues, when you start in a lab in a master's setting or in a PhD setting or even a professional setting, you'll often hear your supervisor, your mentor say, now what's the story?
Yeah.
What's the story with the paper that you're trying to publish or even the whole thesis behind your whole research program? What's the story? And then as soon as that's decided and you focus on that, bam, now all the interpretation happens according to this story. And problems can arise. So I appreciate you outlining that.
Yeah, I think that's one of the problem is, not to get too deep into this topic, but the problem is like all the experimental results are usually like we have to acknowledge that this is an incomplete model of reality. We are trying to model some situation which may or may not be, may have predictive power. So some of our colleagues are working on these ideas where you should examine the merit of the model based on the predictive power. So we think that sort of the mitochondrial perspective on cancer, just the metabolism itself, I wouldn't even go into the origin of cancer. And most people also we work on prevention a little bit, but prevention is not the primary focus of our lab. We are focused more of after you get a cancer, what do you do? Not like how to prevent cancer, which is difficult. So ignoring carcinogenesis itself, but if you look at the bioenergetics of tumors, we think when you change variables in different models, you can predict better what will happen in the real world. Whereas in somatic mutation theory, I feel that's sort of the primary flaw that I would say it has. It's like sometimes it makes predictions that happen, but other times it doesn't. And that's sort of the problem. And another issue is like, and in the lab, as we were discussing, the secret is like you can technically design experiments to confirm your hypothesis. Like if you have knowledge on the variables,
of course.
Like if I wanted to show that oxidative phosphorylation is the primary fuel for a cancer cell, I can very easily design that experiment. I can put them in galactose for 6 hours. I can treat them with some mitochondrial inhibitor like oligomycin or rotenone or cyanide, or put them in hypoxia, and they will die immediately. So in that situation, oxidative phosphorylation was their primary fuel source because you were forcing them to use oxidative phosphorylation. But that doesn't tell you anything about sufficiency or insufficiency or the translational sort of value of that. So I, as I said, I often try to focus on like long-term experimental designs, and in the clinic, I think we have to move more into sort of shared proactive involvement of the patient in the treatment process, and actually be adjusting variables and treatments based on the responses, not sort of just blindly just monotherapy treatment and after you fail that treatment, let's do another monotherapy treatment, and after you fail that one, like that's I don't think that's the approach. I think it should be like, I don't like the word integrative. I think it has bad connotations, but it should be just building from the biology of the human, of the mammal, of the eukaryotic cells, the collection of eukaryotic cells that you have in front of you, trying to like optimize the health of that system rather than sort of just throwing darts at targeted therapies or chemotherapy or something like that.
You mentioned prevention. Prevention obviously is ideal. Is there any evidence to suggest that a ketogenic diet does help prevent cancers?
On the mechanistic side of it, I would say yes. Now, of course, you will always find, people that are allegedly, like, this is mostly epidemiology, so it's like very hard to say anything. But if you have a person that has eaten like a high carbohydrate diet their whole life and they are fine, but you have other person that do the same thing and they, develop obesity and low grade inflammation and they develop a cancer, I think from the basic perspective, yes, a ketogenic diet, low carbohydrate diet with low insulin signaling and low inflammation would be sort of on first principles, even from the somatic mutation theory, I would say the best sort of prevention strategy as compared to the Western sort of standard American diet where you're putting so much energy in the system, and it's the wrong kinds of nutrients and the wrong kinds of signal to the cells, and you are basically so overwhelming the system. And I think this in the long term will damage mitochondrial function invariably in some population of cells, and it predisposes them to develop cancer from both perspectives. Somatic mutation theory perspective, you could say, oh yeah, well, the inflammation reduces the fidelity of DNA replication, and that causes problems, and you develop these mutations and you have cancer now. Or from the mitochondrial metabolic theory perspective, yes, you chronically damage your mitochondrial efficiency, and you now are more predisposed in usually as we usually say, like in epithelial cells or stromal cells, you can, these are the cells that can undergo the metabolic switch to become malignant.
Now, what would your, coming from your perspective as a clinician and as a researcher, what would your advice be to clinicians regarding the scientific method and integration of these disciplines? And then what would your advice be to researchers to help look at that grander picture?
Yeah, that's a tough question, because you have to like sample the idealism with the reality of the work, because like in an ideal setting, as I said, I think most published literature has one or two, like a Nature paper, a Science paper, a Cell paper, these massive, behemoth papers that appear. I would argue that nobody reads actually, but like nobody looks at the data in detail, maybe except poor reviewers. But these papers usually have like one or two very interesting observations, and then the rest is just elucidation of mechanism, which is important. But in our lab, we focus more on
that's a very good point.
practicality of it. Now, my solution would be to give like 20 years to each group to like actually make a stronger collection of these interesting observations to try to like answer a clinical question, or like the actual translational interest of your program, of your topic, of your hypothesis. Like, don't just study the reductionist model that you have for 20 years, but actually dedicate the time to try to link it from the beginning to the end. If you are studying embryogenesis, okay, how does this apply to actual developmental defects or something like that? If you are studying some signaling cascade, how does this translate into the human? I understand that this is what like most of us researchers are trying to do. It's very difficult to do because you have to like splice it and try to publish these small observations. But I think this is unrealistic. I don't know how we would fund science if it was like, oh yeah, now we're giving 20 years to these guys to like, develop some applicable treatment or some result of their work. But yeah, I think most papers should focus a little bit more on, they should make a better case on the translational value of whatever they are studying.
What are you currently working on in the lab?
Yeah, as I mentioned, I've been working on basically this very basic characterization really of like cancer bioenergetics compared to normal cell bioenergetics in my models, which I acknowledge that they might not represent all tumor cells, neither all normal cells. And you would think that this has been done before, but not really. Like, as you mentioned, like cell culture media was developed, like Harry Eagle and others, Dobeko. It was sort of standardized in the beginning of research, sort of cell culture models. And after that, it was designed to support optimal proliferation. So you would have the most amount of cancer cells to study without ever representing the normal bioenergetics or the normal metabolism or asking any questions about the metabolism. So I'm trying to like go back and actually interrogate normal cells and cancer cells to answer the question, what pathways they are using for ATP production without looking at other, like where do they get their ATP? And I think this, other people are working on bioenergetics, but I think this sort of necessary legwork and going through the literature, I have not found as much as you would expect if this was sort of the primary objective, because as you know, like metabolism like faded a little bit after the discovery of the DNA and all these molecular biology studies that focus more on like the signaling and the genetic alterations, the transcriptomic data, rather than basic metabolism. So I'm trying to investigate more in detail these observations.
Now, as we wrap up here, what would your advice be? And I ask this question of every guest. What would your advice be to students in particular, whether they're undergraduate students or maybe pursuing graduate work, who might find themselves working on a controversial subject, pressure from multiple areas? What would your advice be to them?
Yeah, that's another sort of insightful question, I think, but I don't know if my answer will be satisfactory. I feel that's like asking the captain of the Titanic what makes like a great captain and what advice he would give to others. I feel I'm in a very privileged position because I'm actually doing the type of research that I always wanted to do from the beginning with a lot of freedom. But as we mentioned, there's a lot of sort of misunderstanding and a lack of communication, I would say, with other researchers in the field. And the funding is an issue because funding agencies and peer review within funding, you are being read by your colleagues, and your colleagues, if they have preconceived notions of what they would like you to do, they don't, they have like different goals. So if your goals do not align with their goals or their technicality, like techniques and things you should study, then there is an issue. I mean, in our lab, we focus on the treatment as the primary outcome. So it would be great for us to like fund a study where we would just test a bunch of drugs, like inhibitors or metabolic inhibitors or different composition of diets, to just focus on like therapeutic efficacy. Okay, can we cure? We shouldn't use that word. Can we achieve long-term management of different tumor models, expand the tumor models, and expand the therapeutic arsenal that we would use in the preclinical side or in the cell culture sort of in vitro side, sort of understanding more about the bioenergetics. Or I also had this grant that I'm applying to. We'll see. Doing the hybrid experiments, so the nuclear cytoplasm transfer experiments, which have been done by several groups, but not in the, this was in the past, in the 80s, and not really looking at the genetic defects. So we actually don't know what sort of mutations those cells had when they switched the nucleus and the cytoplasm between the cells, and they observed that the nucleus does not induce tumorigenesis. The nucleus with the mutation, if you put it in a normal cytoplasm, from a normal cell, it did not induce mutations. And if you do the opposite, which is very difficult to explain from the somatic mutation theory perspective.
Very counterintuitive.
Yeah, if you take the cytoplasm of a malignant cell and you put the normal nucleus in, and then you inject those cells into this was like a rat model, like a neonatal rats, they develop tumors. This should be important. Like, there's no way, even like with epigenetics, I think it's very hard to explain. That was sort of the explanation, but like cytoplasm of a cell should not contain anything permanent that would induce a malignant, a tumorigenic model with a normal nucleus. If you assume that the nucleus has the genetic material, then this is like very difficult to explain. So these sort of experiments I was proposing in this grant, I was met with sort of like, what are you talking about? Like, we all know that mutations are not necessary to cause cancer. I was like, really? Do we know that? I thought this was like a very interesting observation, but maybe I don't know. These are anonymous reviewers, but so if that's the case, if we like accept that mutations are either necessary or sufficient to cause tumors, why are we spending billions of dollars developing targeted therapies and developing immunotherapies based on like neoantigens or epitopes caused by mutations, if we all agree that mutations are like a downstream effect of the tumor itself, but not primary to transformation? So I think there's like a little bit of disconnect between our perspective and the perspective of the field. So I think maybe what I'm doing right now is like try to communicate better these ideas is what will be necessary to that would be my advice. Like if you are working on a topic that you feel passionate about, you also have to sort of be empathetic and put your mindset into other colleagues in the field to see what they think and try to like explain as best as you can your perspective. So that is sort of what I would say.
Dr. Tomás Duraj, this has been absolutely fascinating from a molecular biologist perspective. I've learned a lot, and you've got me thinking and doubting some of the stuff that I've done in the lab in the past. Where can our viewers get more information on your lab and anything that you might be doing on the side?
Yeah, so I'm a postdoc at the CFIT lab. So I would be remiss to say that if people want to support our work, they can look at our Boston College faculty profile, biology profile, where they could donate to support our work. We get most of our funding through philanthropy and private foundations, but of course always trying to apply for grants in standard NCI perspective. And for myself, I have like a personal website, and I have Instagram and some of these social media profiles, so people can always find me there. I'm always happy to answer any question, be it molecular or clinical, and people can support me that way and get in touch with me that way.
It's been an absolute honor. Thank you, sir.
Thank you, Tim.